system, where we're going to deal with the brain, the brainstem, and the spinal cord. Okay, so we begin with the brain, right? The brain has four major regions: the cerebrum, diencephalon, brainstem, and the cerebellum. Okay, they look a little different, and they're in different locations. All right, and what they are, they're collections of tissue called gray and white matter. All right, gray matter are unmyelinated axons, neuron somas, and dendrites, and white matter are myelinated axons. Right, the white matter regions are sometimes referred to as tracts. Okay, the gray matter in the brain and the central nervous system is designed for signal processing, while the white matter is designed for signal transmission. The difference between the two: signal processing is where we take an electrical signal that we've received from a nerve bringing information into the central nervous system from some other location in the body. We modify it before we send it to an area of white matter, right? And signal transmission is simply moving the electrical signal from one location to another. Okay, so whenever you see white matter in the central nervous system, think superhighway. And whenever you see gray matter, think information processing center. Okay, the cerebrum has two hemispheres with 5 lobes per hemisphere, and the outer surface of the brain has ridges and grooves in it. Right, the gyri are the ridges, and the sulci are the grooves. The reason for these features is to increase the surface area of the brain, because the brain has to do a lot of electrical work in a tiny volume. All right, and that's been true of a lot of other organ systems in the body for quite some time. You'll see, for instance, that modifications to increase surface area are in certain tissues, right, such as the microvilli on simple cuboidal cells on the apical end. Okay, or in the digestive system, the fact that there are ridges and grooves and finger-like projections on the inner lining in order to increase area for absorption and secretion. All right, so this is a repeating pattern, right? When every time you see folds and grooves, think most of the time it's the result of an increase in surface area, right? Some terms we want to become familiar with. Moving towards the anterior of the central nervous system is termed rostral, which means literally towards the nose, and posterior is termed caudal, which means towards the spinal cord. Okay, you can see the brain here. All right, we can get the laser pointer going here, and you'll see here the temporal, the occipital, and the parietal lobe, and then up here is the frontal lobe. All right, and then there's another lobe, a fifth lobe, called the insula, which is actually embedded underneath this lateral sulcus. So we would have to pull those apart in order to see it. And each of the lobes have gyri and sulci on them, but they also have different jobs. Okay, they perform functions that are the same in most individuals. All right, there are some exceptions. In certain individuals, parts of the brain do other types of work, but in most individuals, the frontal lobe is for reasoning, thinking, will, and the generation of motor activity. Okay, the parietal lobe is to process and label somatosensory information. That's stimuli that comes from the surface skin and our joints and our muscles, as well as in some cases special sense organs. Right, the temporal lobe is to deal with balance and hearing. Okay, and the occipital lobe is to deal with vision, and then that fifth lobe, the insula, is to deal with taste and what they call visceral sensation. Okay, so the front side of the brain here is the voluntary command center and the thinking part, and the back side of the brain behind this groove, okay, which is called the central sulcus, is to deal with stimuli that come in and label it, and then move that information to the frontal lobe of the brain so we can decide what we want to do about it. Okay, The cerebellum down here is designed to make sure that our movement intentions and our movement actions match up, okay? So it takes information in from our special senses and our general senses, and it compares that to our intended movements, and if they don't match, the cerebellum will fix that information and send that back up to the frontal lobe of the brain, and then that information will head down again to the cerebellum, but also to the brainstem and the spinal cord, and ultimately out to peripheral nerves, where it'll talk to muscles and we'll do our movements, okay? You can see the underside of the brain here, all right? You can see some of the cranial nerves as well. This is the olfactory nerve here and here. This is the optic nerve, okay? This is the optic chiasm. The mammillary bodies are back here. This is the pituitary gland. This is the pons, and this is the medulla oblongata. These things that look like a couple of baseball bats, okay? And you can see some temporal lobe here, frontal lobe here, okay, a little bit of occipital lobe back here. There's cerebellum, okay? And you'll notice even in the cerebellum, right, you've got grooves and ridges, and that's, again, to increase the surface area. And then here we have a sagittal cut through the brain where you can see the corpus callosum, the septum pellucidum, the fornix, the thalamus, which is the brain's router, the cerebellum, the arbor vitae, which is this tract of white matter running through here, the hypothalamus, the infundibulum, which is a little stalk right here, and the pituitary gland, right? That's the optic chiasm where the optic tracts cross and then give off the optic nerves that go into the back of each eyeball, okay? And then behind this membrane, the septum pellucidum, is a hollow space called the lateral ventricle. There is a third ventricle here between the two thalami, and a fourth ventricle here between the cerebellum and the pons. And the purpose of the ventricles is to produce and circulate a liquid called cerebrospinal fluid, in which the entire central nervous system floats in order to protect it and keeping it from bumping against the hard outer casing that it's in, right? The brain and the brainstem are inside the cranial cavity, and the spinal cord is inside the vertebral column, right? And they can't be bashing up against those hard surfaces because they would get damaged, okay? The brain, in addition to floating in the cerebrospinal fluid, is also tethered to the floor of the cranium as a result of attachments of membranes called meninges, which surround and protect the entire central nervous system. There are a set of membranes from outer to inner. They are the dura mater, the arachnoid, and the pia mater, okay? And what they do is they protect the brain, but they also tether it in place so it's not smacking against the hard protection that the brain has around it. And the same is true with the, we're going to learn with the cord. The spinal cord tethered vertically by something called the filum terminale, and it's tethered horizontally by something called the denticulate ligaments. That's to keep the cord from bobbing up and down and left and right too much. So we just want to kind of hold it in place and let it float in that nice cerebrospinal fluid. Traumatic brain injury is brain damage that results from trauma, which is what? Concussion, blunt force, penetrating wound, okay? The most common type is a concussion, where we see temporary loss of consciousness, headache, drowsiness, confusion, maybe amnesia. And sometimes this can result in damage that can't be repaired, and that can have an effect on the functioning of the brain. It can change the intellect, the personality, and the mood, right? Contusion is a bruising of the brain due to trauma, okay? And then sometimes we have something called second impact syndrome, which is a second injury that occurs before the first injury resolves, and it develops as what we call a swelling of the brain. And that can be dangerous because it can force the brain through the foramen magnum and damage that tissue, okay? Or it could force the brain against the cranial cavity and damage the tissue that way by crushing it, right? The brain, it begins development in the 3rd week of embryonic development, and what we have there is in the 3rd week a thickening along the embryo called the neural plate, which is induced by a rod of tissue underneath it called the notochord, okay? So the notochord produces chemical signals, and that causes the ectoderm layer, which is the top layer of the embryonic disc, to thicken in that region. And that is eventually going to become a neural tube and drop below the level of the ectoderm and become what will be your spinal cord, your brainstem, and ultimately your brain, right? The neural plate becomes a neural groove before it becomes a neural tube. Cells reproduce along the lateral plate margins and become neural folds, and the tips of those plates become neural crests, and they migrate out to different structures in the body and contribute to things like the facial bones and some of the special sense organs, okay? As neurulation continues, the neural groove deepens as folds rise and approach each other, and then the crest cells separate from the folds, and then the tips of these folds will seal, and that will become the neural tube. So by the end of the 3rd week, the folds meet and form the neural tube, which surrounds the neural canal, all right? Fusion begins in the middle and progresses towards the caudal and rostral ends. Openings at the cranial and caudal ends of the tube, called neuropores, normally close by the end of the 4th week, okay? So it's kind of interesting that the way that the nervous system develops seems to reflect the way the nervous system developed in evolution, right? Which is that the first thing really to show up as far as a central nervous system was a spinal cord. The next thing to show up was a brain stem, and then the last thing to show up was what we think of as a brain, but what we technically call the cerebral cortex. And that probably indicates that the memory of evolution is retained in our genes and is simply repeated every time a new person is born, okay? For a short time, the neural tube is open at both ends so that we have a cranial and caudal neuropore, but if the openings fail to close, then we'll have what's called neural tube defects, okay? And this seems to be linked to a lack of B vitamins in the diet of the mother, and we don't understand the mechanism for that. And those can include things like anencephaly, which is a baby born without a brain, and spina bifida, where portions of the spinal cord are actually outside the body, okay? And each of these can present in different levels of severity, okay? Believe it or not, with anencephaly, okay, you, even with the complete absence of a brain, sometimes have the infant survive after birth, okay, because they have a functioning brain stem, which is enough to allow our vital organs to function and our autonomic nervous system to operate, okay? And so they would have the central nervous system equivalent of something like a reptile, okay? Obviously, they can't learn very much and they can't be taught very much, all right? But it goes to show you that in terms of you being alive, your brain stem is much more important than your brain, all right? And that's because your brain stem controls your vital functions. If your brain stem is destroyed, you die, okay? Whereas you can theoretically get by without a brain. Spina bifida is the failure to close the caudal portion of the neural tube, and it can present as spina bifida cystica, where you will have almost no formation of a vertebral arch you generate a large cyst in the back, and that can cause paralysis and infection. And you're often wheelchair bound. We have to fix this surgically, okay? In spina bifida occulta, you have a partial defect of bony arch, and you end up with not nearly as severe a condition. And occulta means hidden, okay? So it can present in different levels of severity. And we can pick this up on ultrasound, okay? The brain vesicles are going to begin to form by the late 4th week in the cranial end of the neural tube. The forebrain is termed the prosencephalon, the midbrain the mesencephalon, and the hindbrain is the rhombencephalon. From the 5th week, the telencephalon is going to develop into the cerebrum. The diencephalon remains the diencephalon and becomes the structures known as the thalamus, hypothalamus, epithalamus, and subthalamus. The mesencephalon will become the midbrain, which is going to connect the diencephalon to the medulla oblongata, and the metencephalon becomes the pons and cerebellum, while the myelencephalon becomes the medulla oblongata. Okay, so in terms of what shows up, right? The medulla is going to show up before the cerebellum, which is going to show up before the midbrain, which is going to show up before the diencephalon, and the last thing to develop is up here. It's the telencephalon, and that again is mirroring the development of the nervous system in evolution. Okay, the brain continues to develop in the fetus between 13 and 26 weeks. The telencephalon envelops the diencephalon and generates surface folds. The folds let the brain fit within the cranial cavity. Most of the gyri and sulci are present at birth, and again, their purpose is to increase the surface area of the brain. So I know it seems a little odd that we make this comparison between evolution and development, but if you Google the brain of a frog or an alligator, okay, what you'll see is something that looks a lot like a human brain stem with very little cerebral cortex, okay? And that's sufficient for those creatures to operate. You really don't see cerebral cortex until you get up to the higher mammals, okay? And even in the more primitive mammals, the cerebral cortex isn't very extensive, and we're going to see that when we do a sheep brain dissection in the upcoming labs. You'll see that the brain is rather small, and the amount of cortex is much less than in a human brain, and that reflects, number one, the difference in size of the two organisms, but it also reflects that a sheep doesn't have to do as much with its brain as a human does, okay? So the folds let the brain fit within the cranial cavity while retaining all that surface area, and allows it to do all of that work. And then we discussed gray and white matter earlier, right? In the brain, the gray matter is on the surface, known as the cortex, and the white matter is in the core with little spots of gray matter embedded in some of the white matter, okay? And these are called nuclei. The white matter are the myelinated axons, and they are for signal transmission. And you can see here the gray matter on the surface and the white matter in the core. Now, the cortex of the brain is very interesting because the most recent neurons in the cortex are actually on the surface, while the older ones are closer to the white matter, okay? And that represents the fact that as the cortex develops, as it thickens, the newer neurons climb on top of the older ones and then begin to do the work of signal processing, okay? So it's the opposite of the way a tree is, right? When you cut through a tree and you see the tree rings, the oldest rings are on the outside and the youngest rings are in the core. Well, this is the reverse of that, okay? You can also see the tracts of white matter. The corpus callosum is a tract of white matter that lets the two hemispheres of the brain communicate with each other, right? And then you've got association tracts that allow movement of information between lobes on one side or other of the brain. And then finally, you have bridging tracts that bring the information down from the brain to the brainstem and the spinal cord. Okay, so those are the superhighways that are present in the brain. Okay, you can see the cerebellum and brainstem here. Again, notice gray matter on the outside, white matter on the inside, and you'll see a continuation of the ventricles of the brain in the central canal. Okay, this is the cerebral aqueduct here, but that's going to lead into the fourth ventricle, and that will lead into the central canal, as well as leading into what we call median and lateral apertures. Okay, so you've got some inner gray matter, you've got some outer white matter as we transition from the cerebral cortex to the brainstem. This is at the level of the medulla oblongata, and then by the time we get to the spinal cord, you'll notice that the gray matter is completely inside and the white matter is completely outside, right? So in the spinal cord, the superhighways are on the peripheral part of the spinal cord, and the processing is in the core. Okay, so we start with gray on the outside and we end up with gray on the inside. Okay, now the meninges of the brain are designed to protect it from chemical and physical insults, right? These are 3 layers of connective tissue that support the delicate brain tissue. They enclose and protect blood vessels that supply the brain with oxygen and nutrients, and they also aid in the circulation of cerebrospinal fluid by allowing it to re-enter the blood at a region called the dural venous sinus, which is a big blood-filled area between the 2 cerebral hemispheres. So as we go from deep to superficial, the pia mater actually clings to every convolution of the brain and has a lot of blood vessels in it. The arachnoid mater is above it and has web-like extensions and has a lot of cerebrospinal fluid circulating around it in an area called the subarachnoid space, which is between the arachnoid mater and the pia mater. And then the dura mater is a tough, fibrous outer covering that anchors the central nervous system to the surrounding bony structures so they don't bob and weave and smack against other structures too much, right? So there's the pia mater, right? Innermost meninge, thin layer of areolar connective tissue, lots of blood vessels, right? And then the arachnoid is above the pia, right? Trabeculae are the web-like extensions that from the pia to the subarachnoid space, which is filled with cerebrospinal fluid. The web is made of collagen and elastin, and it lies under the dura mater, while the subdural space is a region between the dura and the arachnoid that can fill with blood if we blow a blood vessel. Right? The dura mater is the tough outer membrane made of dense, irregular connective tissue, and in the brain it has a meningeal layer, which is the deep layer of the dura, and a periosteal layer, which is the layer of the dura that lies just under the periosteum of the bone. Okay, and so in the brain, it is a double layered membrane, and between those 2 layers is the dural venous sinus, and that has a lot of blood inside it. Right. The epidural space is a space between the dura and the skull. Okay. And it can fill with blood in the event that we have a hemorrhage in that region. Okay, so you can see the meninges here, right? This would be the skin of the scalp. Okay, there's periosteum, there's cranial bone, there's more periosteum, and then underneath you got dura, right? There's your dural venous sinus. These structures are called arachnoid granulations, and that's where the cerebrospinal fluid, which is produced from filtering the blood in ventricles, rejoins the blood and goes back into the circulation, right? So there's a constant flow of CSF into and out of the central nervous system without any buildup of pressure. And then you've got down here the subarachnoid space, and above that the arachnoid mater, and the CSF is in here. And then clinging to the convolutions of the brain, we have the pia, right? And you see all these blood vessels with the pia just underneath them, okay? So the brain is well protected, and that's probably the reason that the neurons in the central nervous system have lost the ability to reproduce and repair. Okay, over evolutionary time, the evolution dictated that these structures are so well protected that they didn't need the extra ability to regenerate. The neurons in the peripheral nervous system have some ability to repair themselves, right? If you clip a nerve fiber, sometimes it can regrow and find its target again, right? But if you do that in the central nervous system, it doesn't grow back. And there's a variety of reasons for that. An epidural hematoma is a pool of blood in the epidural space of the brain, usually because of a severe blow to the head. We call that blunt force trauma, right? Adjacent brain tissue is distorted and compressed and can lead to severe neurological injury or death unless bleeding is stopped and the blood is removed. A subdural hematoma is a hemorrhage in the subdural space coming from ruptured veins that result from things like rotational head movement and can cause compression of brain tissue and usually is slower than an epidural hematoma. Now, the reason we worry about these is that two things are going on here. Number one, wherever the bleed is, everything downstream of bleed is getting starved of blood and could die, right? The other problem, more immediate problem, is the buildup of pressure in these areas that can crush the brain tissue and irreversibly damage it, all right? So it's one of the reasons that we worry so much about things like concussions, because you can bleed into your brain and that can be lethal, okay? Now, the central nervous system is so well protected that it has virtually no immune capability. The only immune capability in the central nervous system, as we learned from the nervous tissue lecture, are the microglia, all right? So if anything gets into the central nervous system through what we call the blood-brain barrier, which is created by the ependymal cells that line the ventricles and the central canal, and the astrocytes that are sort of the middleman between the blood supply and the neurons, then we have a real problem, because we don't have the ability to really fight the infection. In addition to that, if the immune system from the rest of our body moves in through that break, it can destroy the nervous tissue because it's never seen it before, and it will consider it an enemy, okay? In meningitis, we have inflammation of the meninges caused by viral or bacterial infections. We see fever, headache, vomiting, and a stiff neck. Pain from the meninges is sometimes referred to the back of the neck. That's an example, again, of referred pain, and can result in brain damage and death if we don't treat it, right? Bacterial meningitis has even more severe symptoms. That will be bacteria actually alive in the cerebrospinal fluid, okay, as well as in the meninges. Encephalitis is an inflammation of the brain which can be caused by viral infections, and we see things like drowsiness, fever, headache, neck pain, and can also result in death, okay? So basically, your central nervous system has to be protected from the outside world, or else it really doesn't have any way to defend itself. The cranial dural septa are sheets of dura mater extend into the cranial cavity, and they form partitions between the brain areas and also anchor the brain and provide support for the brain. The falx cerebri is the largest of the septa and is located in the midline and projects into the longitudinal fissures between the cerebral hemispheres, and contains the superior sagittal and inferior sagittal sinus. And the tentorium cerebelli separates the occipital and temporal lobes from the cerebellum and has a transverse sinus within its posterior border and a straight sinus on the mid-sagittal plane, and a notch in the anterior surface that allows room for the brain stem to function. Okay, so what are these? These are areas between the double layered dura, okay, that are going to contain blood, all right, and are also going to serve to anchor and stabilize the position of the brain and the brain stem. And we'll actually see those in our sheep brain dissection. We'll remove them. The cranial dural septa also include the falx cerebelli, which runs vertically in the mid-sagittal plane and separates the right and left cerebellar hemispheres. And the diaphragm celli, which is the smallest septa and forms a roof over the sella turcica of the sphenoid. Remember that the sphenoid houses the pituitary gland in the pituitary fossa. Okay, and this is why frequently when we remove a brain from an animal, we tear off the pituitary gland because it gets stuck, it gets jammed in that roof and in that pituitary fossa, right? So all you have left is the infundibulum. So you can see the dural septa here, right? So they're supporting the brain, they have blood in them, they stabilize the position of the brain, okay? The brain ventricles are hollow cavities within the brain that are lined with ependymal cells and are areas where the cerebrospinal fluid is circulated and generated, okay? They're connected to each other as well as to the central canal, and they're also connected to the subarachnoid space, okay? So that's all one big complex looking chamber, right? The ventricles in the brain include two lateral ventricles, which are in the cerebrum and are separated by that membrane called the septum pellucidum. A third ventricle, which is a space in the middle of the diencephalon between the two thalami of the brain. And there is an interventricular foramen that connects the third and the lateral ventricles together. And then a fourth ventricle that's connected to the third by the cerebral aqueduct, and it opens to the subarachnoid space both medially and laterally, as well as into the central canal of the spinal cord. So you can see here the hollow cavities inside the brain. That's a hollow cavity, right? That's another hollow cavity. That's a hollow canal. That's a hollow cavity. And that's a hollow cavity. And then there's all that subarachnoid space all around the outside of the entire central nervous system that's also connected to these ventricles, right? Via the fourth ventricle. Okay? And so that's what it looks like from the front, right? So these look like backwards Cincinnati Reds logos, right? And then this looks a little bit like a crow's head, okay? Cerebrospinal fluid is a clear, colorless liquid surrounding the central nervous system, and it circulates in ventricles and in the subarachnoid space. And what it's there for is to provide the proper chemical environment and proper pH and the proper temperature for the neurons to do their work and for the glial cells to function. And it also provides a way to float the structures of the central nervous system, okay? And so it's constantly made and reabsorbed in the central nervous system, right? It's formed by the choroid plexuses in the ventricles of the brain, which are basically collections of capillaries that are surrounded by ependymal cells where we can force a component of the blood into the ventricles, which we term the cerebrospinal fluid. If we compare CSF to plasma, there's more chloride and less potassium, calcium, and glucose, okay? So there's some selectivity going on by the ependymal cells. In addition, the ependymal secretions and interstitial fluid from the subarachnoid space make up CSF as well, right? So it's continuously formed in the ventricles, but it's continuously reabsorbed by what are called the arachnoid villi, sometimes called the arachnoid granulations. And what they do is they protrude into the dural venous sinus, and that's how the CSF gets back into the blood. If that didn't happen, the pressure from the generation of cerebrospinal fluid would end up crushing the delicate brain tissue. Okay, so you can see what the choroid plexuses looks like here, right? These are the ependymal cells, right? These are the capillaries, and then this is the space. This might be a ventricular space, for instance, okay? And that's where the CSF would first appear. The flow is relatively straightforward, right? We go from either of the lateral ventricles into the third ventricle, down the cerebral aqueduct into the fourth ventricle. From the fourth ventricle, we go down into the central canal, but we also exit the median and lateral apertures and circulate around the entire outside of the central nervous system. And then we reabsorb into the blood at the dural venous sinuses, at the arachnoid granulations, which you can see on the diagram, and you can see here in close-up, okay? So as long as that continues to occur, we're in good shape. But sometimes children are born that have an impingement on that circulation, okay? So there's a block somewhere, and the result is a buildup of cerebrospinal fluid that causes the cranial vault to expand so that it looks a little bit like a light bulb. And this is something that we can't allow to go on, all right? So what we do is we put a shunt into the ventricle where the block is, and we drain that cerebrospinal fluid down into the abdominal pelvic cavity where it gets reabsorbed by the blood vessels in the peritoneum, okay? And that causes the pressure in the cranial vault to go down, and then the cranium can assume its normal size, okay? So this is a birth defect, and we really don't understand why it happens, but it does. And that shunt has to be left in for the rest of the child's life, okay? The reason that this doesn't result in the death of the child very often is because in a newborn, the cranial bones have yet to seal, and so they can expand because of the connective tissue between them, and that's why the vault enlarges. If this happens in the adult, it can be lethal within minutes. That's why we have to leave the shunt in. The blood-brain barrier is generated again by the astrocytes and the ependymal cells, right? So it regulates the substances that get into the interstitial fluid that bathes the central nervous system, and it protects the neurons from exposure to harmful substances that are water soluble, that are hydrophilic, right? So those could be charged molecules, they could be polar molecules, okay? Remember, though, that some molecules aren't going to be blocked by the blood-brain barrier and will pass right through it because they are hydrophobic. Alcohol is an example. Certain drugs like anesthetics can do this as well, okay? And this is one of the reasons why we have to be careful around lipid-soluble compounds, because they're not restricted in terms of where they can go, okay? Specialized capillaries contain endothelial cells that are connected by tight junctions and have a thick basement membrane wrapped by the perivascular feet of the astrocytes, and they in turn wrap other sets of feet around the neurons, okay? The choroid plexus is where the production of cerebrospinal fluid occurs. The hypothalamus and the pineal gland are critical here because they need to secrete hormones directly into the bloodstream, okay? So that is a connection between the central nervous system and the blood supply, and it is a vulnerable spot in the central nervous system sometimes where pathogens can sneak in, okay? Because it needs to monitor conditions in the blood. You can see what the blood-brain barrier with the astrocytes looks like here, okay? And we've already seen how the ependymal cells establish that barrier. Now, the cerebrum is the thing that you think of most often when you think of a brain, okay? It's the wrinkly outer covering of the brain. It's the origin of all complex intellectual function. It's the origin of our will, our personality, our logic, and it's also where we generate motor action plans, okay? These are large hemispheres on the superior aspect of the brain. The cerebrum also processes special and somatosensory stimuli, okay? And sends it to the frontal lobe so that we can decide what we want to do about it, right? So this is really where all the signal processing is going on in the cerebral cortex, okay? Cerebral hemispheres are a paired mass on either side of the longitudinal fissure, okay? At a few locations, there are tracks that connect the hemispheres, and the big one is the corpus callosum. That is the big communication superhighway between the right and the left hemisphere of the brain. An unusual aspect of this is that the right half of the brain controls the left half of the body and vice versa, and most of the stimuli obey that as well, right? So stimuli from the right half of the body are processed by the left hemisphere of the brain and vice versa, right? And you might wonder, well, why is that? And the honest answer is we don't know, okay? It just is. There are animals that aren't set up that way. There are animals where the right side of the body, right side of what is their brain that's the way it goes, right? That's the way they're hooked up. Insects are that way. But humans have this, it's called decussation, where commands and a lot of sensory information cross from one side of the nervous system to the other. Some higher order functions can exhibit what are called lateralization, being that they're controlled mostly by one side of the brain. An example is speech, which is often located in the left cerebral hemisphere in an area called Broca's and Wernicke's region. Okay, so most people, that's on the left side. Now, that's not hard and fast, right? Some people, it may be in other places, okay? The reason that we have to take what we know about the brain with a grain of salt is because the brain is the one organ in the body, and I guess we can include the brainstem, that you can't take out, work on, and put back, okay? And so we know less about it probably than any other organ in the body, even though it's super duper important, okay? And so everything that we know about the brain comes from either brain damage or animal models, and those are not perfect, okay? So when we tell you what we know about the brain, it's with a certain degree of certainty, but it's not 100%, right? You can see the different lobes here, right? Frontal, parietal, occipital. We can't see the temporal on the side. We see the insula, which is beneath the temporal and the parietal, okay? And even though the two sides of the brain look the same, right, the work that they do is different, okay? The lobes of the cerebrum do different jobs. There's 5 lobes in each hemisphere, and they're named for the cranial bones on top of them, right? So there's a frontal lobe, the frontal bones on top of that, parietal lobe, the parietal bones on top of that, occipital lobe, the occipital bones on top of that, and temporal lobe, and the temporal bone is on top of that. And then the 5th lobe, the insula, is it's beneath the parietal and the temporal lobes, okay? The frontal lobe is the front part of the cerebrum. The posterior border is the central sulcus, and right in front of the central sulcus is the precentral gyrus, okay? And that's the area where the motor cortex is located. That's where finalized motor action plans get sent before they go down to the brainstem and the spinal cord, right? The lateral sulcus separates the inferior frontal lobe from the temporal, right? And the frontal lobe, as we've discussed, is where the formation of our motor plans happens. That's our discussion with our skeletal muscle. It's where our higher thought and reasoning are located. It's where our will is located, okay? And it's where our personality is located. That's why if you damage it, you see big changes in those functions, okay? The parietal lobe is behind the frontal lobe and in front of the occipital lobe, okay? The anterior border is the central sulcus. The postcentral gyrus is a ridge just behind the central sulcus, and the posterior border is the parieto-occipital sulcus. The lateral border is the lateral sulcus. So what does the parietal lobe do? Well, it receives somatosensory information, which comes from the surface of our skin, our muscles, our joints, okay? And in some cases, some of our special sense organs, all right? And then it takes that information and it attaches meaning to it using memory areas that are behind the somatosensory cortex. And then it takes all of that information along with special sensory information and combines that into a signal that it sends to the frontal lobe of the brain, where we decide what we want to do about what we're experiencing. Okay? The temporal lobe is located under the lateral sulcus and is important in hearing, balance, and in olfaction and smell. Okay? The occipital lobe is on the back side of the brain, and it's important in vision and in visual memory, while the insula is involved in visceral sensation and taste. Okay? The motor areas are housed within the frontal lobes. Okay? So the primary motor cortex is on the precentral gyrus and controls skeletal muscle activity in different locations of the body. And we can generate a map called the motor homunculus that shows us what parts of the cerebral cortex control what part of the body. Okay, what part of the motor cortex is talking, for instance, to your legs, your feet, your trunk, your hands, your fingers, your face, and so on. And what you see in the primary motor cortex is that the proportions of surface area of the brain dedicated to different parts of the body is not in proportion to our body proportions. And that's because in certain areas of the body, we need more motor control, such as your hands, your fingers, your muscles in your face, your tongue, and so on, okay? Whereas in other regions of the body, you don't need nearly as much, such as your arm, your legs, your trunk, okay? And so this represents two things, right? The amount of surface area of the brain dedicated to that, as well as the amount of information coming in from those parts of the body, right? So you've got more nerve endings, for instance, in your hands and your fingertips than you have, say, in your trunk, okay? And so more area on the brain surface is dedicated to that, okay? And we know this because we can actually, in healthy individuals, stimulate these parts of the brain, and we see those parts of the body move, okay? So this is a piece of information about the brain that we're pretty certain about because we can test it on healthy individuals. All right. So other areas of the cerebrum include Broca's area, each, right? And this is on the left side of the brain in most people, okay? And that controls movement involved in vocalization. The frontal eye field, which is on the superior surface of the middle and frontal gyrus, and it regulates eye movements for reading and binocular vision. And the premotor cortex, which is in front of the motor cortex, and that's where we actually plan our motor activities. So that's like a staging area for what we want to do, right? So that's the part of the brain that says, okay, maybe I should move my arm along with my legs and my fingers. And then once we decide on that, we send that to the motor cortex, and then it heads down to the brainstem and the spinal cord and the cerebellum, okay? All right. The primary somatosensory cortex is behind the central sulcus in the region called postcentral gyrus, and it receives somatosensory information from the surface of the body, our muscles, our joints, and in some cases, some of our special senses, okay? And so we can, just like we did with the motor cortex, we can make a sensory cortex model of the body that's pretty similar, okay? Somatosensory association areas are behind the primary somatosensory cortex, and that's where we assign meaning to the stimuli that we pick up from the postcentral gyrus, okay? So we use memory to give perception to our sensations that arrive at the postcentral gyrus, right? So when we're talking about a stimulus, it's always sensation first and perception second, right? Sensation is just something's happening, and perception is what exactly is it that's happening to us, and that's shaped by our memory. And so you can see here the somatosensory cortex. Both of these are frontal sections through the brain, right? And so, this would be the right side, this would be the middle, this would be the left side. You can see again that you've got a lot more sensory nerve endings in your hands and fingers and face and lips and tongue than you have in places like your trunk, your legs, and your feet and your arms, okay? And that represents, again, two things, right? There's more nerve endings coming in there, and so more surface area of the brain has to be dedicated to that, to processing all of that extra information, okay? The primary visual cortex is at the tip of the occipital lobe of the brain, while the association areas are in front of that extreme tip. And the way that this works is that the sensation of vision arrives at the primary visual cortex, and then the visual association area uses memories to attach meanings to what you see, okay? Primary auditory cortex is in the temporal lobe, kind of on the top, and the association area is sort of underneath that, and that works the same way, right? You hear something, you get the sensation of sound, and then you attach meaning to that using the stored memories that you have, okay? So the memories are critical to your experience in the world, okay? The primary olfactory cortex is also in the temporal lobe and provides conscious awareness of smell. And it's the only sense that doesn't go through the thalamus on its way to the cerebral cortex. And the primary gustatory cortex is in the insula and provides information about taste, which is actually a combination of both our gustatory cortex information and our olfactory cortex information. Okay. Then there are integrating portions of the brain, right? The prefrontal cortex is rostral to the premotor area, and that's where complex thought, judgment, personality, planning, and decision making happen, and is still a developing area in adolescence, and it's also an area that gets damaged through alcohol and recreational drug use, okay? And so that's why we worry about people that use a lot of recreational drugs, because you're impeding the functioning of the prefrontal cortex, and that's going to affect your judgment, okay? Wernicke's area is located in the left hemisphere and is involved in the comprehension of language. So that's the sensory part of speech, right? What am I hearing and what do I want to say? And Broca's area is how do I say it, right? Okay. Autism is a condition in which we're unable to interpret or express our emotional content with regard to the world around us, okay? It has a spectrum of presentation. It's more prominent in males than females, okay? It really, we really don't know what causes it, okay? There's, I'm sure, genetic and environmental components, but we don't really have nailed down a reason that this occurs. Basically, what this is this is a disconnection of your emotional brain from the rest of your brain, okay? Your emotional brain is formed by a set of structures that are sort of around the corpus callosum, okay? And this is collectively known as the limbic system, all right? The limbic system involves dealing with memory, dealing with odor, dealing with the ability to express and detect emotion, all right? And in individuals where the limbic system either isn't hooked up or isn't working right, then you can't express things like anger or sadness or happiness, and you can't detect them in the actions and the facial expressions of others, okay? So the emotional content of your world is gone, all right? So an example, right? You might smell donuts cooking, right? And you'd say that smells good, right? But somebody with autism might smell the same thing and say that smells like donuts, right? So they wouldn't have an emotional component to their experience. They still experience it, but there wouldn't be any feelings about it, okay? Now the white matter, as we already discussed, is involved in moving information in the central nervous system from one location to another. It's deep to the cerebral cortex and is composed of association, commissural, and projection tracts, right? The association tracts are in one or the other hemisphere, and they move information from one lobe in a hemisphere to another lobe, or within a lobe to another location in that same lobe, right? Arcuate fibers are the short tracts that connect neighboring gyri and longitudinal fasciculi, or longer ones that connect gyri in different lobes, okay? And then the commissural tracts are between the hemispheres and bring information from the left to the right and the right to the left. So you can see those here, right? The commissural tracts in the white matter and the corpus callosum, and then these association fibers here, right? Now this is all white matter. These are all superhighways, okay? And then there are projection tracts, right? And the projection tracts bring the information down from the cerebral cortex to the brain stem and the spinal cord, okay? And they pass through a region between the thalamus and the cerebral nuclei called the internal capsule, right? And you can see here what the projection tracts look like. They're highlighted in green. And you'll see that in a lot of these projection tracts that you cross over in an area known as the medulla oblongata, a location called the decussation of the pyramids, all right? And basically, that's that funky thing about the central nervous system that states that in human beings and in higher animals, right, the right side of the brain controls the left half of the body and vice versa, okay? And we really don't understand why that is. It just is, okay? Patalis is a protrusion of a lobe on one side compared to another. Right-handed individuals tend to have right frontal patalis and left occipital patalis, and left-handed individuals tend to show the opposite, okay? We're one of the few species that shows dominance on one side or the other, right-handed versus left-handed, etc. Okay, and that's literally because of a difference in the two hemispheres of the brain, right? There's a difference in the number of connections in one hemisphere versus the other, and that's why a lot of people are right-hand dominant, for instance. Now, there's other things that can lateralize as well, okay? Cerebral lateralization is where two sides of the cerebellum show differences in higher order function. Categorical hemispheres are usually on the left and are specialized for language, categorization, and analysis. We call it your logical side of the brain. And representational hemisphere is usually on the right, and we might call that your artistic brain, okay? The two hemispheres are constantly talking to each other through the corpus callosum, okay? Which, incidentally, is different in the two sexes, okay? The corpus callosum is more extensive in females than in males, all right? And that results in differences in the outcome of things like a stroke, okay? So if a man has a stroke in a region of the brain, he's more likely to completely lose a set of functions that he's learned than if a woman has a stroke in the same location, because of the more extensive corpus callosum and the reduction in lateralization of those functions, right? So in a female brain, you split the stuff out more often. In a male brain, it's on one side or the other more often, okay? And so that means that your stroke recovery is going to be different, okay? And it's not that one setup is better than the other, it's just that they're different, okay? So you can see some examples of lateralization in this diagram, okay? You'd see how functions in the right and the left hemisphere are not mirror images of each other, right? You've got musical ability and recognition of faces on the right side, and you've got language and math comprehension and speech on the left side in most people, right? That's not hard and fast because there is a property that neurons have called plasticity that allows them to move information around in the event of damage to some extent. Epilepsy is a neurological disorder where neurons that generate action potentials do so inordinately quickly, okay? It can be controlled by medications, but sometimes we have to do surgery, okay? In severe cases, we might have to do removal of a hemisphere, which is the removal of a side of the brain responsible for the seizure activity, while leaving the corresponding region in the other hemisphere intact, okay? And so the remaining hemisphere takes over the function of the missing hemisphere. And again, why does this happen? We don't really know, okay? There seems to be both a genetic and an environmental component. Now, in a cerebrovascular accident, what we see is either a lack or a reduction of blood supply to a portion of the brain. It can be because of a ruptured or a blocked artery, okay? And it can cause brain tissue death if it goes on for more than 5 minutes, because that's what we call your oxygen reserve in the body. Symptoms of blurred vision, weakness, headache, dizziness, and problems walking can result, and it will always affect the opposite side of the body where the damage took place. A brief episode is called a TIA, where you have a very short-lived reduction in blood flow, and it can present as trembling, loss of muscle control, loss of sensation, but it usually resolves. It usually comes back, okay? The cerebral nuclei are just deep layers of gray matter embedded in the white matter of the brain, okay? So you've got cerebral cortex, which is all gray, and then under that you've got the white matter tracts, and then there will be little pockets in the white matter tracts of gray matter, and those are the nuclei. And what they do is they regulate your motor output, all right? The caudate nucleus has a head and a tail that parallels the lateral ventricle, and it produces pattern of rhythm and walking movements. The lentiform nuclei is a rounded mass between the insula and the diencephalon made of the putamen and globus pallidus. Lentiform means lens-like, and this is because it looks like a lens if you cut it in a coronal section, okay? The putamen helps control movements at the subconscious level, and the globus pallidus influences the thalamus to adjust muscle tone. Basically, the function of these guys is to serve as gatekeepers to determine when a motor action plan is allowed to come down from the cerebral cortex to the brainstem and the spinal cord, okay? That's one of their big functions, okay? So they're like gatekeepers. Sometimes they shut the gate, right? And sometimes they open the gate, and then we can do our movements, okay? The claustrum is a sliver of gray matter immediately internal to the insula cortex that processes visual information subconsciously, while the amygdaloid body is an expanded region at the tail of the caudate nucleus important in mood and emotion. And the corpus striatum is a striated area in the internal capsule. You can see these different nuclei here, okay? Again, basically what they're doing is coordinating movement, but also allowing the motor commands to descend, right? To go from the cortex to the brainstem down here and the spinal cord below it, okay? Okay, headaches can be due to dilated blood vessels in the skull or due to muscle contraction that constricts blood vessels. A migraine headache is a severe recurring unilateral pain. It's not a true brain disorder, but can accompany a brain disorder. Uh, Cerebral palsy is an umbrella term that basically refers to trauma to the brain. Okay, it results from damage to the infant brain before, during, or immediately after birth, and we see impairment of skeletal muscle control, and we see sometimes cognitive deficit. Okay, inability to learn. All right, learning problems. Huntington's disease is a hereditary autosomal dominant, meaning it's on chromosomes other than the sex chromosomes, and you only need one copy for the disease to show up. Okay, it results in rapid, jerky, involuntary movements, intellectual deterioration, basically neuron death, and it's fatal within a decade or two of its onset, and it usually begins to show up in mid to late adulthood. Okay, so the tragedy of this disease is that we can detect it uh, prior to birth, right, and we can detect it after birth. Um, but a lot of times, what will happen is that children will find out that one of their parents has Huntington's, and they may or may not carry the gene for the disease. And if they carry the gene for the disease, they're going to die of it. And so they often opt not to test to see if they have the gene because they want to live the balance of their life um, without that hanging over their head, okay, and deal with it when and if it occurs. Parkinson's is the result of the death of the substantia nigra of the midbrain. The substantia nigra of the midbrain produce dopamine, okay, and that helps to um, control and coordinate movement and also allows ease in beginning and stopping movement, okay. And so when people have Parkinson's, they have problems with uh, fluid movements and initiating and stopping movements. It does not impair their intellect. Okay, and again, we don't know why these neurons die. They just do. Okay, we have had some success with repopulating those neurons in animal models of Parkinson's disease, and we've done some work in humans. Okay, and have seen some recovery in cases. Um, and the reason that we can do this is because the brain has immune privilege, right? You can work on the brain, and as long as you don't break the blood-brain barrier, you don't have to worry about the phenomenon of tissue rejection. Okay, so you can remove pieces of the brain or even put in material and not worry about the immune system coming in and destroying it. Right now, we will pick up um, with the diencephalon in the remainder of this chapter, and that will be posted on the website. Okay, we're at the midway point for 13 at the moment, so I'm going to cut it here, and I will see my Monday group uh, in lab. All right, and we will discuss the nervous system, and then the remainder of this is going to be put up as a podcast. Okay, the diencephalon is an area above the midbrain that includes the epithalamus, thalamus, and hypothalamus. The names give you an idea of the location, right? The hypothalamus is below the thalamus, and the epithalamus is above the hypothalamus, right? Using the prefixes as their location. Different functions, right? The thalamus is the brain's router, so that the stimuli that are coming from the skin surface, the muscles, and the joints gets routed to the right region of the cerebral cortex. Okay. The hypothalamus is the overlord of the endocrine system and the control center for the autonomic nervous system. Okay. And the epithalamus contains structures that are connected to the limbic system, which is our emotional brain. Okay? The epithalamus forms the posterior part of the roof of the diencephalon and covers the third ventricle. The pineal gland is an endocrine gland that secretes melatonin and helps regulate the day-night cycles, which are circadian rhythm. And it's going to lie 180 degrees opposite the location of the pituitary gland. So it's on the back of the brainstem, at the level of the diencephalon, as opposed to on the front, right? The habenular nuclei relay signals from the limbic system to the midbrain and are involved in visceral and emotional responses to odors. Okay, so these are again part of the limbic system, right? Pineal gland's role with melatonin is to work on opening the gates of perception depending on light levels, right? So the way it works is that as light levels go down, that information from your eyes, from your retina, is able to walk back to nerve leads that talk to the pineal gland, and that results in the release of melatonin. And what that does is that begins to close off um, the, um, the ability of the thalamus to let stimuli get up to the cerebral cortex, and so you become less aware of what's going on around you, and you fall asleep, right? And then as the light levels come up, melatonin levels go down, and the reverse happens. There's also a very important collection of neurons called the reticular activating system that plays a role in this as well. And that's basically a column of gray matter that runs across the dorsal part of the brainstem. Okay? The thalamus are oval masses on top of the midbrain and lateral to the third ventricle. They're connected by something called the interthalamic adhesion, which is gray matter that connects the two so that the two sides can talk to each other. They're composed of thalamic nuclei. Now, what are these? These are nerve cells that have a particular task. Okay, this is the third use of the word nuclei in this class, right? The nucleus of an atom is where the protons and neutrons are. The nucleus of a cell is where the DNA is located. And nuclei in a tissue are collections of cells in a tissue that have a particular task. Okay, so the axons from a given nucleus are going to project to a particular region of the cortex and bring the information to it. Okay, it receives all signals from conscious senses with the exception of olfaction, which goes straight to the temporal lobe, and it relays those signals to the appropriate area of the cortex and filters out stuff that we don't need to pay attention to. You can see the location of the different nuclei here. Now, if you were to look at an actual thalamus, you wouldn't see this, right? It would all look like a mass of gray matter, okay? But the bottom line is that it serves as a way to get the stimuli that are deemed important to the region of the cortex designed to allow us sensation of the stimulus and allow us to draw on memory so that we have perception of the stimulus. The hypothalamus is underneath the thalamus and above the pituitary gland. Okay? It's the anterior-inferior region of the diencephalon. A stalk called the infundibulum is going to extend from the hypothalamus to the pituitary. So it rests in the pituitary fossa of the sphenoid bone, which is in the middle of the sella turcica, right, but is attached by the infundibulum to the hypothalamus. The hypothalamus, as we've discussed, is the overlord of both the endocrine and the autonomic nervous system, okay? As the operator of the autonomic nervous system, it's going to control things like heart rate, blood pressure, and so on, okay? Endocrine system activity is to control the pituitary gland, which in turn controls other endocrine organs. And so there's two lobes of the pituitary. The posterior lobe is an actual outgrowth of the hypothalamus. It's literally neural tissue. And the anterior pituitary is glandular epithelium, which secretes hormones designed to control other endocrine organs as well as hormones that can control things like our growth rate. Okay. It also is important in the regulation of body temperature. It's your body's thermostat. Okay. Um, it controls hunger, right? Ventral medial nuclei monitor nutrient levels and regulate when we want to eat. It controls water intake because anterior nuclei monitor the concentration of solutes in the blood, and that regulates thirst. And it's also important in sleep-wake cycles because the suprachiasmatic nuclei are going to direct the pineal gland to make melatonin, and that's going to control our circadian rhythm. Suprachiasmatic just means above the optic chiasm, which is the area where the nerve fibers that ultimately become the optic nerve cross from right to left and left to right. Okay? So this is part of the relay system that gets light information back to the pineal gland for the purposes of controlling melatonin levels, right? And then there are emotions that are here, okay? It's an important part of the limbic system that responds to things like pleasure and fear. So what you really have in the hypothalamus is kind of like a second brain, okay? It's a little teeny thing. And it controls everything necessary along with the brainstem to basically keep you alive, all right? All the other stuff, which is superficial to the thalamus, isn't that critical to our survival, okay? It's critical for higher thought, reasoning, interacting with your environment, and so on, okay? But in terms of just existing, you could get by with the diencephalon and just a little teeny tiny bit of cortex, and that's a lot like what a frog brain or an alligator brain look like, okay? So this is just to show you that the aspects of the nervous system that were critical to survival are the more primitive ones. They were the ones that showed up first, okay? And that's why they're housed here in the hypothalamus, which is why if you lose hypothalamus function, you're going to die, okay? The brainstem, which is below the diencephalon, connects the cerebrum, the diencephalon, and the cerebellum to the spinal cord, and is made up of ascending and descending and decussating nerve tracts that bring information up, down, and left to right on its way either up to or down from the brain. Okay, it has autonomic nuclei. It also contains nuclei of cranial nerves. It also has reflex centers in it, and then the three parts are from superior to inferior: the midbrain, the pons, and the medulla oblongata. And you can see a picture of what the brainstem looks like here. Okay, if we go to laser pointer, all right, what you'll see here, this is thalamus, right? All right, these are the mammillary bodies. This is the optic chiasm. These are going to become the optic nerves here. Okay. Then you've got the optic tracts leading back. Okay, so these are all part of cranial nerve anatomy. All right, and then here is the midbrain. Okay, this is the pons. This is the medulla oblongata, and then all of these yellow leads that you see coming out, those are cranial nerves, and we'll learn about those in a bit. Okay, they serve structures in the head and in the neck. All right, most of the brainstem is white matter. All right, but there is some gray matter in there, and those are those nuclei that we talked about. This is what it looks like from the side. Okay, so you can again see here the pineal gland, right, the thalamus. These are the colliculi. These are the superior and inferior colliculi, collectively called the corpora quadrigemina, and they're part of the midbrain. All right. These are the cerebral peduncles, and these are just tracts that bring information up to the thalamus. And then you've got the pons, which is connected to the midbrain. It's connected to the cerebellum. It's connected to the medulla oblongata. Okay, it's basically a bunch of tracts. There's a few nuclei in here that control things like your breathing rhythm. Okay, and then down here is the fourth ventricle, right? That little chamber there, and then down below this is the medulla oblongata. Okay. So if we start with the midbrain, right, the cerebral peduncles are just motor tracts on the anterior lateral surface of the midbrain that carry voluntary motor commands from the primary motor cortex down to the medulla oblongata through the pons, right? The superior cerebellar peduncles connect the cerebellum to the midbrain, and the medial lemniscus are bands of ascending myelinated axons that run through the brainstem. Remember, white matter is for signal transmission. The substantia nigra of the midbrain are dopaminergic neurons that are important in movement, emotion, pleasure, and pain. And its degeneration, as we discussed earlier, is one of the causes of a condition known as Parkinson's disease. The tegmentum is involved in postural motor control and contains the red nuclei, which get that color from the extensive vascularization in the region, as well as the reticular formation, which is involved in sleep and wakefulness. Right? The cerebral aqueduct connects the third and fourth ventricles and is surrounded by periaqueductal gray matter and houses the nuclei of the oculomotor and trochlear nerves, which talk to muscles that move the eyeball. Okay? The tectum contains four mounds making up a tectal plate. The superior colliculi control visual reflexes and tracking with your eyes, and the inferior colliculi control auditory reflexes. So here you can see the midbrain, right? There's the red nucleus. There are the dopaminergic neurons of the substantia nigra, right? There's the peduncle, right? Cerebral peduncle, and you can see the aqueduct here, and there are the colliculi on the back, okay? And there's your reticular formation. We are talking about that earlier, right? That's one of the things that talks to the thalamus in addition to the melatonin levels to open the gates of perception, right? That's why we wake up or fall asleep. The pons is underneath the midbrain and above the medulla oblongata, and it's made up of white matter tracts, right? The middle cerebellar peduncles are transverse axons that hook the pons to the cerebellum. The pontine respiratory center regulates the muscles of breathing, along with nuclei in the medulla oblongata, and the superior olivary nuclei help with sound localization. The cranial nerve nuclei that are found here include cranial nerves 5, 6, 7, and 8, which are the trigeminal, abducens, facial, and vestibulocochlear nerves. And again, we'll talk about what those nerves do in a bit. And there's the pons, right? Mostly white matter, right? Some gray matter scattered throughout. Okay, again, reticular formation continues through the pons and into the medulla oblongata, right? So you can see all these structures here. Okay, and this is what it looks like from the side. And again, remember that the yellow leads are cranial nerves. The medulla oblongata is the most inferior portion of the brainstem and is continuous with the spinal cord. It has the pons above it. And on the front, it has a pair of baseball bat-shaped structures called the pyramids, which are where the descending tracts, the motor tracts, cross from right to left and left to right at a region called the decussation of the pyramids. Decussation means to cross over. The olives are bulges that are lateral to each pyramid and contain the inferior olivary nuclei, which relay proprioreceptive information to the cerebellum. Now you might wonder, what's proprioreceptive information? That is information about your body's position in space. Okay? And then the inferior cerebellar peduncles are tracts that connect the medulla to the cerebellum. Okay? The cardiac center of the medulla regulates the heart rate and the force of contraction. The vasomotor center regulates blood vessel diameter and has an influence on blood pressure. And the respiratory center controls your breathing rate, with the dorsal and the ventral respiratory groups communicating with the pontine respiratory group in the pons. Okay, and so what this does is this allows you to control your breathing rate when you're unconscious, right? Or when you're not actively thinking about your own breathing, Okay, you can control your diaphragm both consciously and using the brainstem when you're asleep. Other nuclei include regions for coughing, sneezing, vomiting, salivating, and swallowing. Right? Cranial nerve nuclei in the medulla include the vestibulocochlear, glossopharyngeal, vagus, accessory, and hypoglossal nerves. The nucleus cuneatus and gracilis. The gracilis is information for the lower limbs, okay, and the cuneatus is for the trunk and the upper limbs, okay. So that's somatosensory information that's coming through those, and they send signals through the medial lemniscus up to the thalamus, and then the thalamus is going to send that information on to the correct area in the cortex. All right? So there is the cross-sectional view of the medulla, and again, notice mostly white matter with gray matter scattered throughout. Okay, and you've got a lot of cranial nerves in there. And there's the lateral view where you can see the olives and the pyramids. Okay, and the cuneatus and gracilis, right? And the respiratory nuclei as well, okay? Okay. Now the cerebellum is the second largest brain area after the cerebral cortex. It has a cortex that has ridges and grooves in it, okay? The surface folds are called folia, and there are hemispheres, a left and right cerebellar hemisphere. Each hemisphere has an anterior and posterior lobe separated by a primary fissure. And there's a region called the vermis, which is a narrow band of cortex between the left and right lobes that receives sensory signals regarding the torso and the overall balance of the body. The three regions of the cerebellum are the cerebellar cortex, which is the gray matter on the outside, the arbor vitae, which is the core of white matter that appears like a branching tree, and deep cerebellar nuclei, which are, of course, gray matter. Okay? Three nerve tracts connect the cerebellum to the brainstem. The superior cerebellar peduncles are going to connect the cerebellum to midbrain, the middle peduncles to the pons, and the inferior peduncles to the medulla oblongata. And you can see cerebellum here, right? So there's fourth ventricle, right? There's cerebral aqueduct. That's central canal. That's pons here. There's medulla oblongata, okay? And that's arbor vitae, that branching pattern here, okay? And the gray matter on the outside with the folia, okay? And then here's what it looks like from the top, right? So there's the left and the right lobe, and then right there is the vermis, okay? What does the cerebellum do? Well, it coordinates movement and it makes sure that our motor intentions match our motor actions, okay? It can store memories of previously learned movements. It regulates activity along voluntary and involuntary motor paths and adjusts movement that is initiated by the cerebrum, making sure that our movements are smooth. It helps us maintain equilibrium and posture and receives motor plans and sensory feedback. So basically what's happening here, this is the kind of the schematic, right, is that when we generate a motor action plan and we want to bring it down, right, it comes down and then a copy of it goes to the cerebellum, and then the rest of it is going to come down through the pons, through the medulla oblongata, and down to the spinal cord, and ultimately out peripheral nerves to muscle targets, okay? And then, as information comes in from our eyes and our balance apparatus, our vestibular apparatus, okay, and also our muscles and our joints, our proprioception, then we compare that to what we received from the cortex, right? So this is ascending information in blue here coming up, and that's for comparison to the red information, right? And then if there's a change, if there's something goofy, right, then we correct the information we got initially and we send that back up to the cortex, and then that modified plan is what shoots down, shoots across again, and then goes down to the pons and medulla oblongata and the spinal cord. So it's our sort of on-the-fly movement editor, okay? People that have damage to the cerebellum, one of the things that you see is a condition called cerebellar ataxia, in which they have a hard time with a smooth gait, and they have a hard time initiating and stopping their movements. Okay? Alcohol will have an effect on all brain activity, but it definitely hits the cerebellum. Um, Alcohol that can soak through the blood and into the brain causes a problem with gait, balance and posture, and a problem with picking up proprioceptive information. That's why when you get drunk, you kind of stagger and fall over. Okay. Now, the limbic system is our emotional brain. It's what allows us to perceive and to express emotional content in our experiences. Okay. What are the components? The cingulate gyrus above the corpus callosum, the parahippocampal gyrus, which is in the temporal lobe and is associated with a structure called the hippocampus, which is sort of like the body's DVR. Okay, the hippocampus receives stimuli during the day that we think of as conscious memory, and then at night when we dream, it's putting that information into the appropriate memory locations in the brain so that we can draw on them later. Okay? The amygdaloid body is involved in aspects of emotion and memory. Fear is one of the emotions housed in the amygdaloid body. Olfactory bulbs, tracts, and cortex are basically for odor, right? Which is a strong evoker of memories. And then the fornix is a white tract that connects the hippocampus to the other limbic structures. Okay, so it's kind of a commissural tract. Nuclei that connect to other parts of the limbic system include the anterior thalamic nuclei, habenular and septal nuclei, and the mammillary bodies. And you can see the limbic system structures here. Okay, there they are. Okay, they all occur sort of around the callosum and thalamus. All right, they're a little superior and lateral to it. Okay, and they are, if functioning properly, we have emotional content and richness in our lives. A disease that affects the limbic system function is autism. And in people who are autistic, one of the hallmarks of a lot of autistic individuals is that they cannot perceive or express emotions in their daily experience, okay, because something is wrong with the development or the interconnections of the limbic system, okay, and we don't understand why autism happens. The reticular formation is organized gray matter in the brainstem. It's a dorsal column of gray matter, and it regulates muscle tone through spinal cord connections. It assists in autonomic function through brainstem connections, and it's one of its big roles is in the reticular activating system, where it's able to, through connections with the thalamus, either open or close the gates of perception, and that controls our alertness. Okay, so we talked a little bit earlier about how this operates, right? Light levels go down, you pick that up in your retina. That information goes back not only to the occipital lobe of your brain, because you can see that it's getting dark, but it also goes back to the pineal gland, and melatonin levels start to rise. And in addition to that, right, that is going to impact the communication of the reticular activating system to the thalamus, and that'll shut these gates down that lead to the cerebral cortex, and you'll fall asleep. Okay, and then when light levels go up, the melatonin levels go down, and then the reticular activating system opens the gates of perception, and then we're aware of the world around us more effectively. Okay, so that's one of the reasons why sometimes people with sleeping problems will take melatonin in order to regulate that. Okay. Some examples of states of unconsciousness that are abnormal include fainting, because of usually inadequate blood flow to a region of the brain in the cerebrum. Stupor, which is means you're only arousable to extreme stimuli, and this is often the result of liver, kidney disease, brain trauma, drug use, or certain metabolic disorders. And then coma, where the sleep from which you do not wake, right? And this is usually trauma, metabolic failure, cerebrovascular accident, stroke, okay? low blood sugar, or certain drugs. And then persistent vegetative state, which is lack of thought and awareness, but non-cognitive brain function will continue, okay? And sometimes you can have spontaneous movement in the persistent vegetative state. There have been tales of people who've been in a coma for years and have awoken from it, okay? So it's very hard to understand in some cases how this comes about and how this stops. We do know that damage to the reticular activating system can induce coma, okay? Higher order functions include things like learning, memory, and reasoning, okay? They're in the cerebral cortex and involve different brain regions with both conscious and unconscious processes controlling them. The development of these functions is more complex as we get older and make more connections between the nerve cells in the cortex. It allows us to move more information around and also to store more information. Okay? Most central nervous system axons are still myelinating during the first 2 years of life, with brain growth rapid in early childhood and 95% complete by age 5. Beyond early childhood, the processing is more complex. New connections get made, others disappear. Some axons are still undergoing myelination in adolescence, okay, so they haven't completely finished their development. And axons of the peripheral nervous system continue to myelinate even past puberty, okay? And remember, the myelination is just an important way to ensure that the integrity of the nerve signal is preserved, right, and that it gets to its ultimate destination. An EEG is just a way to measure the electrical activity in the brain. Electrodes are applied to the cranium, and a readout is produced. They're important in studies of sleep, brain lesions, vegetative states, and so on, as well as the evaluation of a condition known as epilepsy, which includes seizures that can involve blackouts, spasms, and shaking, although not always. Okay? The seizures are termed grand mal and petit mal, depending on the type and the extensive reach of the seizure. Activity occurs in different frequencies. Alpha and beta waves are common when we're awake, and delta and theta are common when we sleep. The electrodes are placed on the head, and then we examine the different types of brain activity, and you can see some of the different waves here. Okay? Now, sleep is a temporary absence of consciousness that's vital to normal brain function. Okay? We think that during sleep, one of the things that happens, in addition to putting our memories in the correct area of the cortex, is that we regulate levels of neurotransmitter. Okay? We see less cortical activity, and vital brainstem function gets maintained as a result of sleep. Sleep stages are characterized by brain waves as well as eye movements. In non-REM sleep, we see slow frequency brain waves that take up 75% of your sleep time, and that's where we get our growth, our rest, our energy conservation, and our strength renewal. It's divided into different stages, right? And REM sleep takes about a quarter of the sleep time, and that's memorable dreaming, and that's critical for the storage of our memories, right? This is when the information that was picked up by the hippocampus gets put into the correct cortex in the brain so that we can draw on it later, okay? An example of a readout here of a typical sleep cycle is located here in this hypnogram. You can see the REM sleep and the different stages, right? Going in and out until we awaken in the morning, right? Sleep requirements change throughout our life. Infants need 17 to 18 hours, teens 8 to 10, and adults 7 to 8. Lack of sleep is associated with depression, memory loss, and poor immune function. Insomnia gets more common as we age. That's difficulty falling asleep. And another problem we will sometimes wrestle with is sleep apnea, which are breathing problems during sleep. We treat obstructive sleep apnea with a device called a CPAP machine, which produces positive pressure and keeps the airway open so that we can breathe comfortably throughout the night. The obstructive sleep apnea is risk factors include obesity, pregnancy, and old age. Okay, This is different from central sleep apnea. Central sleep apnea is a failure of the brainstem to innervate the diaphragm, and it often results in death. Okay, and it's one of the conditions we think contributes to SIDS, sudden infant death syndrome. Cognition is the mental process of awareness, knowledge, memory, perception, and thinking, and the association areas of the cerebrum help us do that. Okay, studies of brain lesion patients are helpful in understanding normal function, but they're not the be all and end all. Okay, again, to study something that's damaged, you've already got a problem with your model, right? Because it's damaged. Frontal lobe patients show personality abnormalities, and the frontal lobe function, um, when it's damaged, we see problems with planning and decision making. Okay, damage to the primary somatosensory cortex causes loss of body awareness on the opposite side. So if there's damage on the right, we experience it on the left, and so on. Agnosia is the inability to recognize or understand the meaning of stimuli. The location of the lesion determines the loss. An example would be a lesion in the temporal lobe leading to the inability to recognize the meaning of sounds or words. And so what's going on with a lot of this is it's like you're cutting the connections that allow us to apply the meaning of memory to the sensation, right? And so it loses its significance to us, right? Um, Sensory memory are associations that are based on sensory input. Um, these last for seconds, and short-term memory is seconds to hours, and you usually can remember about 7 bits of information, while long-term memory can be encoded from short-term if the information gets repeated and can exist indefinitely, or it can be lost if we don't access it occasionally. Okay? And so you can see an example of how we deal with our information, right? Stimuli comes in, we have sensation, memory provides perception, and that is our immediate memory. And then we can move that to short-term memory, and this is in a different location in the brain, okay? And then if we repeat or train ourselves, this can become long-term memory, which is in an even different location in the brain. And the reason we know this is because there are people with different types of deficits. Okay, somebody with dementia, for instance, may lack sensory or short-term but have long-term memory, while somebody with amnesia may have short-term or sensory but lack long-term memory. And those areas of damage are in different parts of the brain. Okay, the areas that are involved in memory include the amygdala and the hippocampus. Remember, we always said that the hippocampus is like the brain's DVR. Um, the long-term memory is housed primarily in the appropriate association cortex. Okay? Motor memories are stored in the premotor cortex, and sounds in the auditory association, and images in the visual association cortex. Okay? All right. Okay, so Alzheimer's disease is basically the death of neurons. And so you lose those connections that allow us to move information around and also take advantage of stored memory. So we see changes in mood and behavior, loss of memory and personality, and we don't know why this happens. There's probably both a genetic and an environmental component. We do know that in individuals that show Alzheimer's, that there are beta amyloid plaques and neurofibrillary tangles, but we don't know whether that's a cause or a result of the disease. Okay, and it's hard for us to know because you'd have to be there when those things form and then see the change in behavior of the patient in order to assign causality, right? There's no cure. There are meds that can slow it, right? We can identify it using PET and loss of olfactory sense as early signs of the disease. Okay. Now, amnesia is a different class of defect. Okay. In amnesia, we see partial or complete loss of memory, Usually it's temporary and affects only a portion of your experience. It can be caused by psychological trauma or direct physical trauma, right? The type and degree of recovery depends on the part of the brain that's been damaged. Damage to the thalamus and limbic structures, especially the hippocampus, can disrupt or prevent the formation of new memories, because remember that that's our mechanism for receiving the stimuli and then storing them back in the cortex. Okay. Now, emotion is how we feel about what we experience, right? And this is primarily the job of the limbic system. They're interpreted by the limbic system. Their expression is controlled by the prefrontal cortex and the frontal lobe. The amygdaloid body and hippocampus are critical. If they're damaged or stimulated, the emotions can either be deadened or exaggerating, and learning can be impaired. So you could think of the amygdaloid body and the hippocampus as sort of a volume control for your emotions. Language involves reading, understanding, speaking, and writing words, right? Wernicke's area is the interpretive region, and it lies in the temporal and parietal lobes in a region that's called the operculum, okay? And the motor area for speech is Broca's, and that's in the frontal lobe, all right? And that's how we speak. The primary motor cortex signals the motor neurons to make the speech, okay? So it's not a surprise that our understanding of language would involve temporal lobe and a little bit of parietal lobe here, okay? And the motor portion is going to be firmly in the frontal lobe, okay? Language is going to involve our categorical hemisphere, which, depending on the individual, is either going to be the left or the right, but in most people, it's the left hemisphere of the brain. The representational hemisphere is going to pick up on the emotional content of the speech, right? A lesion to the right hemisphere, opposite Wernicke's area, causes aphrodisia, which is dull, emotionless speech. Apraxia is a motor disorder where the person is aware of what they want to say, but they can't speak it, while aphasia is difficulty understanding or producing speech. It may not produce comprehensible speech, and we may not realize it, and it's often due to head injury or to stroke. So with apraxia and aphasia, what you're looking at is severing of neuronal connections that are critical to producing speech, right? And so we have a problem turning what we want to say into action, okay? Because we can't get it to the motor cortex to do what we want, okay? Or if it's a problem with understanding it, then we probably have problems connecting the memory of language back to Wernicke's area, or connecting Wernicke's area to other memory areas, okay? Dyslexia is an inherited learning disability. We have problems with single word decoding in dyslexics. Individuals have trouble reading, writing, and spelling, and the level of reading competence is below our expected intelligence. Improvement in some individuals with time is seen, and it may be a form of disconnect syndrome where we have problems transferring information through the callosum, right? So the connection is not going where we intend it to go, okay? Now, the cranial nerves are just portions of the peripheral nervous system that talk to our special sense organs as well as structures in the head and neck. And they're numbered with Roman numerals 1 through 12, 1 being closest to the nose and 12 being closest to the spinal cord, okay? You can see the different cranial nerves here, all right? And we're going to go through each one and talk about what they do. We'll begin with the olfactory nerve, which provides our sense of smell, right? It's the shortest cranial nerve. It runs through the cribriform plate of the ethmoid bone to the olfactory bulb, and the olfactory tract heads straight back to the temporal lobe of the brain, and that's how we interpret smell, okay? So it is entirely a sensory nerve, right? It's only going to have incoming information, right? Optic nerve is another sensory nerve, and it runs from the retina of the eye back to the optic chiasm, back to the lateral geniculate nucleus of the thalamus, and then there are extensions that head all the way to the occipital lobe of the brain. Okay, and this is how we see. Right? This is what delivers vision to the occipital lobe, and then the association area is going to attach meaning to what we've observed. The oculomotor nerve is going to control muscles that move the eye, lift the eyelid, and change pupil diameter as well as lens shape. So it moves through the orbital fissures and talks to the following muscles, right? It's going to talk to the superior and the medial rectus and the inferior rectus, right? And it's also going to talk to the pupil and control the pupillary diameter via a parasympathetic influence, right? So when parasympathetics hit the pupil, what the pupil will do is constrict, okay? So it's an example of a nerve that is mixed in terms of its motor output, okay? The trochlear nerve talks to the superior oblique muscle, okay? And again, it heads through the superior orbital fissure and talks to that muscle so that we can move the eyeball. Well, the trigeminal nerve has three branches that are going to extend to the mandible, the maxilla, and also into the ophthalmic region, okay? And it gets somatic sensation from the face, and it also controls muscles that are involved in chewing, such as the temporal muscle and the masseter, okay? So facial sensation, or we like to call it sensory to the face, and muscles of chewing, okay? And two of its branches are going to move through the sphenoid bone. Remember, the sphenoid bone is the keystone of the cranium, okay? Through the foramen rotundum and the foramen ovale, right? The abducens nerve also goes through the orbital fissure and talks to the lateral rectus muscle and lets the right eye look right and the left eye look left. While the facial nerve is a mixed nerve that is motor to the face. So it controls muscles of facial expression, but it has a sensory component. It brings in taste sensations from the tongue, okay, and it also talks to salivary glands in the region. So again, is a mixed nerve. The vestibulocochlear is responsible for our balance and our hearing, right? So it brings in hearing and balance information into the brainstem, okay, and that allows us to control our dynamic and static equilibrium and also perceive sound, right? The cortex that is involved in that is for hearing, the temporal lobe, and for balance, a region between the temporal and the parietal lobe known as the operculum, okay? Then the organs that do that are the cochlea for hearing and the semicircular canals and the vestibule for equilibrium, and they're embedded in the temporal bone of the skull. The glossopharyngeal nerve is a mixed nerve that gets taste and touch from the tongue and also provides motor control of the pharynx, okay? So it's important in things like swallowing and speech, okay? But it also has a sensory component, okay? So in terms of taste, you've got glossopharyngeal, facial, and vagus all relay taste information back to the brain, okay? The vagus nerve is a mixed nerve that's very unusual because it leaves the head and neck entirely and heads all the way to the thoracic and abdominal pelvic cavity. It controls muscles in the pharynx and larynx, but provides sensation from our abdominal and thoracic viscera, and is also the major parasympathetic lead to those organs, okay? So that's an efferent, involuntary set of commands that goes through that nerve, okay? The spinal accessory is a nerve that controls muscles of the neck and the pharynx, okay? So it talks to things like the sternocleidomastoid and the trapezius, okay? Well, the hypoglossal nerve is a motor nerve that controls the movement of the tongue, okay? And so those are the 12, right? So cranial nerve 12 would be closest to the spinal cord. Cranial nerve 1, as we already know, is literally in the nose, right? And so that's how we number those different cranial nerves, okay? All right, that brings